we are going to start. So you can start those recording. Okay, let me share. Okay, welcome guys. So we start today's lecture, and today is the second day of the pandemics and the review of this current situation and also coronavirus issues. So for starting, so again, we are going to do a little checking of the previous the lecture material. So be ready for this. As you may guess, this the Kahoot will record everything you put in. So it's not clear in the middle, but it records your answer and also your speed and for everything. So try your best. Okay.
교수님, 이 닉네임을 잘못 입력했는데.
That's okay. 채동 we can recognize. That's fine. Someone come quick. Okay, let's start. So we're going to practice some of those numbering. Perfect. Okay, we have a trillion cells in our body. We have a 1조 개 cells in our body. So that's a trillion. So billion is 10 to the 9, million is 10 to the 6, quadrillion is 10 to the what? Quadrillion? Doyun? 10 to the 15, right? That's an upper number. Okay, nice. Good start. One could not answer. So pentakis is 5. It's a penta or pentakis, either way. Pentane, in chemistry, we use penta is 5. Oh, nano. So we practiced a little. I tried to make it clear. Nano is a 10 to the minus 9, right? So nona is a 9. What is a hepta? Hepta? Hey, come quick. Hepta is a 7. Undeka?
11.
Undeka is the 11. What is the 12?
도데카.
Dodeka, nice. How about 10?
데카.
10 was a deca, right? So at least if you can recognize, and that's good. Come on. Penta was easier. Quintet is a 5. What is a 6?
섹스텟.
Sextet, right? 7?
septet.
Septet, was it? Okay. 8? 8 was always easy. It's an octet, right? So oct is easy. Quintet is a 5. Quintet in the music, it can be, quintet is 오중주. Okay. Okay, ternary is one number.
10.
10.
Ternary is 10. Ternary is used in which application? Ternary, usually we use that binary, ternary. Ternary is a 십진법. Ternary, okay. Quinary,
오진
quinary,
5,
it's a quintet, 5. Nonary,
9,
9. Senary,
7.
is it 7 or 6?
6, I guess.
That's a 6. Okay, at least these numbers, if you look at and if you can catch the number, then that's enough. You don't need to memorize, to use this word, but at least if you can understand, that's good enough. Okay, now let's move to a little more practical. Half and half, huh? So lengthwise, which is bigger? Bacteria is bigger, right?
Yes.
Bacteria is bigger. Lengthwise, how many times?
10
10
times
times.
bigger.
10 times. Then if you fill in, that means it's by volume. Then it should be
1000.
10 to the 3, right? So then it should be 1,000, not 10,000. That's clear, right?
Yes.
Yes.
Okay, move on. What a surprise. So 10 to the 10, you can change it into 10 to the 9. Then you can multiply 10 to the 1.5. It should be either 15 gigabyte or you can say 15,000 megabyte. So two of them is a technically correct answer, but which one may I prefer? Which one should we prefer? It should be keep the 15 gigabyte, right? So that's easier to catch. 15,000 megabyte. It has some other better expression we have. So we try to avoid that terms, even though that's a technically correct number, but for easy usage, 15 gigabyte is even easier. Now you guys gave 150 gigabyte and one. Both of them is the same name. Why did you choose one red, one blue? It's my mistake, but anyway, you should avoid this kind of the confusion. Still, that's very difficult. It's a meter. So if you move those the decimal 1, 2, 3, that's a 0.12 millimeter. That's a 10 to the minus 3, right? So technically this one and the next one, 120 micrometer, it's the same. Both of them are maybe acceptable, but we prefer to use a 120 micrometer for this. Okay, we try to avoid 0 point something, or maybe above 1,000. Okay, that's what we are going to try to avoid. This kind of a conversion, it's better. You can convert almost instantly. Then it's quite convenient to catch. When somebody is talking to you with this kind of very big number, very small number, if you can catch that reasonable scale, then that is a very useful ability. So this one, I tell you, you practice and make this is a part of your body. Okay. Whatever you are a scientist, engineer, it doesn't matter. This is a very useful habit. A little more. 2 more questions. Now this is a concentration. Molar concentration. Still that's difficult. You should think whether which direction you should move the decimal, right? You should move to make it smaller or bigger. If it's a micromole, that's a 10 to the minus 6, right? Then 10 to the minus 5 is bigger than 10 to the minus 6. So if you move this into the 10 to the minus 5 to 10 to the minus 6, then you should make 1.5 bigger. Okay, that should be 15, not 0.15. I guess this is not, your ability to do this. This is, the speed, how quickly you can convert this. Okay. Some of you made the wrong answer, either 0.15, that's the wrong direction, and there's a 150 micromolar, that's maybe, how many decimal you should move, kind of mistake. I think you can fix yourself. Okay, just one more, and then we will move on. Still, half of you are getting wrong direction. Do you think you can fix this yourself, or you need some help? Still, it's not clear, then tell me. Then we can practice a little more. This is the last problem. Still confusing? You can do it, right?
Yeah.
But practice. So far, this is a, relatively straightforward. But still, the right answer rate is only half. Half is not acceptable level, but I think this one you can figure out how to do it. So almost the same set of practice, the material was given to you in the last the lecture note, and you practice yourself. This one will continue to practice, but this is a very, very useful, the small thing, but very useful thing that we can do. Okay, we move on. Okay, today, still this week is we are going to study about the general, the features of this coronavirus, and also information collection from other sources. Now here I showed one picture here. This is kind of a representative coronavirus structure. And as you can see here, inside here, this is the genetic information, mainly RNA. But usually when genetic information is packed, RNA is a highly charged molecule. Is it negatively charged or positively charged?
Negative. Negative.
Because of which functional group?
Phosphate.
Phosphate.
Phosphate, right? Phosphate is a strongly negatively charged. So that's why negatively charged material, if it's highly packed, it's not stable. Then usually we need to have counter cation to neutralize those charges. So that's why DNA or RNAs, if they are highly compact structure, they need some partner which can neutralize that charges. Then this N protein should be charged positively or negatively?
Positive.
It should be positively charged, right? So usual protein in the regular cells, which has positively charged, and then bind to DNA and neutralize those charges. Do you know that name of the protein? Nobody knows?
Histone.
Histone.
Sorry, histone. Yes, right. So histone is the protein, positively charged protein, which is binding to DNA and then neutralize the DNA's charges. So in this virus, the name of protein is different. It's not histone, it's N protein, but it should be positively charged and neutralize those the charges. And now here we see envelope, this red color is a protein. envelope is, this red color is this is a lipid, phospholipid bilayer, as in our the cell membrane. And then there is a different proteins, spike protein and M protein, hemagglutinin, esterase. This part maybe you have seen in influenza virus, the video. But anyway, that kind of a protein is embedded inside this membrane structure. This virus has membrane structure uniquely, unlike the other virus. That's why if you use a soap to wash your hands, the soap material is a chemically very similar structure to phospholipid bilayer structures. That's why soap can dissolve this membrane structure. That's why it can disassemble of this protein structure of this virus, and it can destroy. So that's why soap washing hands is a very efficient way to remove this virus. Okay. Now today, I asked you one by one to you whether you wanted to present today. Well, it was ended up with all of you want to present or volunteered. So here are some. We start with this the presentation first today. First one will be Doyun. We'll talk about the current expansion of those virus issues and comparison with the SARS. Someone will talk about the comparison between MERS and COVID virus. And then Juan will talk about the diagnosis of the approach. And Taekjun will talk about the one possible strategy of a treatment. So still this will be general information and also the warming up stage, but you guys selected some complementary topics this way. So remember this order, then you have to start to share your file, and we discuss one by one after you present and discuss it, and then move to the next one. Okay, so this order, remember. So Doyun, you start. Let me start to the sharing my file, and you can share your files here.
Can you see it?
Yes.
Yes.
Okay. Hello, my name is Kim Do-yoon, and my presentation topic is SARS outbreak and COVID-19. Let me talk about SARS outbreak summary first, and then what we've learned from the SARS outbreak to help fight COVID-19. SARS, as you know, is a respiratory severe acute respiratory syndrome. First case came from Guangzhou, China on November 16, 2002. Within months, SARS spread to 26 countries in Europe, North America, and South America, and Asia. The virus spread to over 8,000 people worldwide and killed almost 800. It was far less serious than the present corona disease in the number of infections and deaths. The reaction of China was not so transparent. Initial reaction of China is they were hesitant in sharing information according to this, and it took several months before the Chinese government started sharing information with WHO. No vaccine was developed. However, it came to end on May 18, 2004. What does this date mean? As no new infections had been reported in a 3-week period, WHO announced China as free of further cases of SARS. compared with the SARS 19 years ago. Let me talk about COVID-19 now. COVID-19 is a coronavirus disease with symptoms such as fever, cough, and shortness of breath. First case came from Wuhan, China in December 2019. There is currently no specific antiviral treatment or vaccine for it so far. A new coronavirus disease outbreak 17 years after the SARS outbreak. The new coronavirus SARS-CoV-2, which causes the disease COVID-19, has already surpassed the 2003 SARS outbreak. Questions. Do you think China and the rest of the world have fully learned the lessons from the 2003 SARS outbreak? Maybe it was. First, how do the lessons from SARS to help fight COVID-19? First of all, Chinese government has been much more willing to share information and be open to avoid repeating the mistakes of SARS. Then second thing is about the advances in technology needed to understand the virus and develop diagnostic tests or treatment. In January, Chinese scientists had already sequenced the virus, which first appeared in December. They also made that information available to the scientists around the world. Genetic information about the virus is needed to help scientists develop diagnostic tests for the virus, as well as treatments and vaccines. Several groups are already working on a vaccine. Even if faster vaccine development compared to 17 years ago, it could take 12 to 18 months for a vaccine to be ready for widespread use. Finally, strict public health measures are valid, like the ways that brought the SARS epidemic to an end. Thank you for listening.
Okay, thank you, Do-yoon. So Do-yoon gave some general, don't close it yet, but that's fine, I can do it, don't worry. Do-yoon gave some introduction about the SARS and current situation about the coronavirus. Anybody has any question?
I have a question.
Yeah, go ahead.
It's not kind of scientific question, but anyway, the presenter told the Chinese government is trying to do their best than before. And these days, the government claimed that the situation of the COVID-19 is going to the end in China. Could you believe this?
I guess I didn't understand your question exactly.
Sejin, would you repeat your question?
All right. Anyway, the presenter believes Chinese government is trying to do their best to solve this problem, like sharing their information or like that. And then these days, Chinese government say that this situation, the pandemic of COVID-19 in China is going to the end. My question is, could you believe this government claim?
Well, let me rephrase the questions. So of course, everybody is trying, including Chinese government. Chinese government definitely we believe they are trying to control this situation. That's no question. But the question may be whether their sharing of information is really fair And also those information they are sharing right now or they are claiming right now, is that really some real number we should believe? That's the question, right? So their announcement about their number and situation, is that really something believable? What do you think, Doyon?
I think it's not believable stuff because Chinese government had been hide all information before, so I still believe, not believe.
Yeah, at least in your presentation, you said Chinese learned the lesson and then they are trying their best for sharing information. That part, many people are suspicious. And also their way of counting those impacted other people's number, they use a different criteria. So that part of the diagnosis part, let's discuss after Juan's the presentation. Juan is going to talk about the diagnosis method of the coronavirus. Then we can compare which method is the global standard and also which method is a Chinese way to count at the moment. Okay. Any other question?
Can I give
Sure,
a question too? As we have heard that when the SARS outbreak has come out, there has been no vaccines that have been approved by the FDA or nor been produced. So I wonder why the exact reason or the general cause why a vaccine has not been made, whether it was because the time was not efficient or the SARS outbreak came to a halt before it was effectively being produced. I want to know, like, what the exact reasons why it wasn't produced.
Okay, to answer this question, anybody can try to answer to Dongwoo's question? Why the vaccine is not efficiently developed yet? Why it takes so long time? Anybody try to answer? If you try to answer, then we can figure out, how much you understand and which part you are missing, kind of things. Okay, Juan, try.
May I speak?
Yeah, sure.
Actually, there are a lot of some specific site on coronavirus RNA, as Professor Young-Tae Chang shows before the presentation. But we need to figure out how and what kind of chemicals can attack those kind of sites. But as the other coronavirus before, there are some other sites that some country has officially presented that they are targeting for the coronavirus gene is very different. So it is shame, but we cannot our humans even now human didn't figure out what kind of target gene is the most efficient to treatment this disease. So this is the most important reason why we cannot develop the treatment.
Okay, thank you, Juan. To answer this question, we need to understand what is a vaccine and what is the process of making vaccine and how the vaccine works. Juan gave some technical terms that what can be the target of those vaccine or vaccination. Now we need to figure out, we are talking about that target as a protein or RNA or sequencing. He mentioned whether we are talking about the sequencing of. Now here, when he mentioned, and also the Doyon's presentation, the sequence of a virus was presented by Chinese government. The sequence means sequence of what? Doyon, would you answer? It was your presentation.
sequence of RNA, I guess.
Sequence of RNA, because that is the genetic information of the coronavirus. That's RNA sequence. That RNA sequence implies what? What does that mean, the RNA sequence?
RNA sequence means their information to make a protein.
Make a protein. So genetic information means whether it's our own DNA or own RNA, it doesn't matter. That information is the information to make a protein. That's the genetic information. Then the vaccine target would be, it could be maybe directly on DNA, RNA, maybe, but most of those vaccine target is the protein is the target. Then, based on those sequences, which target we should attack, it was not clearly figured out yet. That's one problem. And then, which part of that, the virus, we should use as a vaccination target. And now we need to understand how we can prepare for it, and until we get approval to use that, what kind of a process that we should take. So those whole process of, at least for the safety test, it takes long time because the test should be done directly on human body. So the test on human body we call clinical test. Clinical test, because of those human right issue and a safety issue, it should be very well designed and it should be performed very carefully. That's why it cannot be done in a month or two. Usually it takes a half a year or 1 year. That is the main issue of taking so long time. Even though there is a possible vaccine developed, checking whether it is safe to use or not, it takes a long time. So whether we can surrogate or avoid that process, that would be key issues. And rumor says, not only rumor, President Trump pushes those scientists. Whatever you say, you just make it out until president election by November. No, we don't have the enough time. Current scientific approach or clinical test, the process-wise, it's impossible, physically impossible time, unless we change totally those process. So there is some issues we can learn more. Those we can understand better after we learn a little more basic other stuffs in due course. Okay, so let's stop here. Let's move on to the next the presentation. Next presenter is Sangwon.
Can you see?
Okay, good.
Yes.
Okay, see that.
Hello, my name is Jo Sung-eun, and I will start presentation about COVID-19. This is the table of contents. Firstly, I will tell you the reason why I chose this topic for presentation. Secondly, general properties of coronaviruses. Third, the differences between MERS and COVID-19 and between influenza and COVID-19. Lastly, I will finish my presentation with consideration of treatment of COVID-19. I chose this topic, COVID-19, for presentation because COVID-19 brought about WHO pandemic announcement, unlike the other coronaviruses, MERS or SARS. So I wonder what are the differences between MERS and COVID-19 and what should be concerned about to develop the treatment of COVID-19. As you know, coronavirus is RNA virus and one of the 3 viruses which cause cough. The RNA in this virus is a transcriptome itself, and they have crown-shaped spike proteins and membrane glycoprotein and envelope protein. The crown-shaped spike proteins combine with the terminals of cells and invade into the cells. There are 4 types of coronavirus. Alpha beta types exert influence on mammals, and gamma delta types exert influence on birds. The other animals' coronaviruses can infect humans through intermediate animals. For example, MERS and COVID-19 is originated from bats, and MERS intermediate animal was camels, and COVID-19's intermediate animals is assumed to snakes or other animals in wild animal market in Wuhan. These are differences of MERS and COVID-19. MERS infect the elderly or chronic disease patients or immunosuppressed people. But COVID-19 is more highly infectious, so it can infect anyone regardless of age. However, average mortality rate of MERS is about 35%, and COVID-19's average mortality rate is about 4%. As you see, MERS was spread out 27 countries, and COVID-19 is spreading out over 150 countries all over the world. So this is why WHO pandemic announcement is only for COVID-19. There are differences between influenza and COVID-19 too. Influenza has vaccine and several options of antiviral drugs, but COVID-19 has no vaccine and rarely and no antiviral drugs specifically. Influenza's incubation period is 1 day to 4 days, and COVID-19's incubation period is longer, 1 day to 14 days. And interestingly, influenza can pass only when patients have symptoms, but COVID-19 can pass on the other people without symptoms. There is no antiviral drug for COVID-19 yet, but there are several treatments that tested for the treatment. First, Calatrava HIV treatment, and secondly, remdesivir treatment. Both treatments are interrupt increase of virus, but these have different principle. Calatrava interrupts the process of HIV viruses invasion into cells, and remdesivir interrupt increase of the viruses by combining with them. Both HIV virus and Ebola virus are RNA virus like COVID-19, so we can assume that this is the reason why we can use this treatment for COVID-19. COVID-19 patients' cause of death is assumed to be cytokine storm, although we can't sure yet. If the cause of death is really cytokine storm, we should stabilize release of cytokine and deter hyperactivity of immune cells. So I think treatment of COVID-19 interconnect with treatment of autoimmune disease. Thank you for listening.
Okay, nice presentation, Son. Any question to Son? There's a lot of information that she gave out. There should be some questions.
I have a question.
Yeah, go ahead.
Why did you suggest the way the interconnecting with the autoimmune disease? I don't know why you suggest the sentence. How it is working?
Cytokine storm is maybe.
So you should explain that term first. Then it can automatically explain why you mentioned that autoimmune disease connection.
Cytokine storm is the maybe over release of cytokine in immune cells. So the immune cells attack the normal cells, not pathogens. So it may be called as the one of autoimmune activity. So
let's define what is a cytokine first. What is a cytokine?
Cytokine is the stuff that released from immune cells.
And what is the function of it?
And
cytokine is released from immune cells, and what does it do?
I forgot the term.
You can use an easy term. You don't need to find out the technical, difficult term. Just use the easy word.
Encourage immune system activity.
Encourage other immune cells. Encourage or activate. Okay. Cytokine, some of them is suppressing other function, but many of those cytokines, main reason of immune system exists is that they fight. To fight with others, they call more of the friends, other immune cells, and activate them. So cytokine storm means a lot of cytokine. Okay, to activate the immune system to be ready to fight against any invader, activate too much. So immune system is overactivated. That is believed was the suspicious about the death of this coronavirus symptom. Then in that sense, this is similar to autoimmune disease. Autoimmune means this is attacking ourselves by overactivated immune system. So the word itself, if you understand those the word, then it can be naturally connected. Cytokine storm means is overactivation of the immune system. That's why if there is any autoimmune disease drug, it may work for this case too, you can assume. Okay? Other question? Okay, let me ask you a question. Someone mentioned about the MERS death rate. Do you remember what percent was it?
36.
36%. Okay. How was the death rate for SARS?
9%.
It was 800 out of 8,000. That's what the Doyon gave out. So 9 or 10%, let's say. So SARS was 10%. MERS was 36%. MERS was much more dangerous, isn't it? But that dangerous thing, when someone presented, only when they had the symptom, now this is influenza, but similar things, MERS has a severe symptom and it has a much higher death rate. So once the patient occurs, then we can notice who is infected. And that person's death rate is too high and very quickly. Then before the virus can spread to other people, the symptom already gave out a warning to other people to isolate that patient. And the patient death rate is very, very high. They die very quickly too. That's why the virus failed to spread quickly. They exposed their identity too quickly. And then they killed their host too quickly too. So in this table, the COVID-19, it has even without symptom, it's kind of a hidden Trojan horse. They are hiding inside. They do not show any sign that the person is infected or not, but they can infect other people. That makes the situation much worse. So I said 10% for SARS, 36% of MERS. What is the current percent of COVID-19 death rate?
4%.
Different countries has a different rate for now, right? So by average, it's about 4%, but depending on how they are treated, Italy has higher than 10%, and maybe lowest is maybe Germany so far, 0.6% with a statistically meaningful number-wise, and the number may increase because the total number, still the patient treatment was not fully done. So let's just say about 1% to 10% range. It's quite broad. Then depending on how we treat the patient, so there is some treatment here, other HIV treatment drugs, or maybe this kind of immune suppressant is kind of a current the treatment method, or maybe preventing other infection. That is called as a secondary occasional opportunity infection. Immune system is down, and overexpression was suppressed by this drug. Then our body is the dependent list. Then other bacteria or other infection can happen. So handling those kind of a symptom is a current treatment, and wait until our body generate our own defense system, so-called antibody. So that's kind of a self-immune system is activated. Until the time we wait, or we just treat the patient just to be healthy. That's kind of a current treatment. So this one we call symptomatic treatment. 대정요법 means it's only handling those symptoms, not the cause. So far, there is no direct drug at the moment. Okay, we have about the presentation. So let's stop here. Let's move to the next presentation. Next one is Juan.
Yes.
Okay, go ahead.
Hello everyone, I am the third presenter this week, Jung-Ju Hwan. I'm a little nervous because previous presenter did very well. I hope that all of you are not be burdened by the fact because I am the student of life science. To begin, let's review the last class presentation. The first presenter, Sun Se-jin, was talked about the research trend for COVID-19. Especially about the treatment of it by using vaccine and medicine. And the second speaker, Chae Dong-woo, shows us a number of visual data and critically analyze the government and by various agencies' migration policy for COVID-19. In fact, it was very impressive to hear about the rebounding, which warned that this cause could spread again while the human race was get off guard after the situation was eased. As I was listening to the accountment, I was wondered how the number of the infected people was being tallied, because the statistical analysis are clearly impossible without the data being collected. And this led to a series of concerns, and then to the question, how can we distinguish between infected and non-infected? So from now on, I'm going to share with you what I learned from my research. Prior to the accountment, I would like to say that I have referred to the procedures currently underway in South Korea. This is a description of the entire process, like the driving through method we've seen in the news. People in the clinic are mostly made up of volunteers. These people collect the samples needed for diagnosis from these who came for inspection, and they then adapt them to preserve them so they can be well preserved. And if you send them to the lab, and the people, researchers, or go through a sample test and decide whether the subject is negative or positive to COVID-19. The sample collection step. Volunteers collect more than 3 milliliters of their spectrum, or kare, from people coming in the cars, and use cotton swab or myeonbong to extract secretion from the phosphorus here inside their necks. If the patient is taken from a hospital, which is not a normal situation, but is obviously a serious case, we take a 5 to 10 milliliter sample of blood with the consent of the patient. So how do we store and transfer these samples? In the case for a cotton swab with the secretion, inoculate it against a strength growth medium called VTM, or virus transport batch. Blood is often stored in a plastic tube called the serum separate tube, which you can see during a blood test in the hospital. These two samples can be denatured by several enzymes in the body, such as apoptosis or so on. So they are refrigerated at around 4 degrees Celsius until 5 days after sampling, and then frozen at about -70 degrees 5 days later. Finally, it is a sample test process that can be also called the core of the diagnostic process. Typically, there are 3 main test procedures using virus diagnosis. The first cell culture test is the easiest step to assess, but it is very dangerous because it cultivates virus without any processing, and it is not inappropriate because it is difficult to clearly identify the result during the diagnosis process. Next, it is a test using antibodies that the professor mentioned in the last class. Today's news occasionally tells us that a device can be developed that can test COVID-19 infections in about 15 minutes or so. Almost all of these devices have been developed using this approach. It doesn't take long to run. It can even diagnose any asymptomatic infections or 무증상 감염자, and also can be used to determine whether it is uninsulated or not. So it is very good way to determine whether people are infected by COVID-19, but however, it has very difficult way for if there is no suitable antibody found. So the WHO doesn't recommend this way because there are no suitable antibody, and in this moment there are about 50% of this test accuracy. So the last way, the molecular assay is now now adapted in these ways. There are two ways that this approach is applied in the beginning of COVID-19, which is pan-corona PCR test, and one is real-time RT-PCR that is being used today. First of all, we using another coronavirus that we've been collecting data from before, like HCOV229E and so on, because we haven't done all of the research in the beginning. We don't have any idea about COVID-19. So using a common diagnostic pan-coronavirus test, that is, the patient is tested by positive for coronavirus infection and then sequencing the samples taken to determine if they are infected with other COVs. If all of the 6 diseases or of the other 6 doesn't match to the patient is COVID infected. The next way is the real-time reverse transcriptase PCR method. If you look at the 5 diagnosis kit here, can you see my cursor?
Yes.
Yes.
These 5 kits are currently officially approved by the South Korean government, and they are manufacturing using this method. I heard that the US Congress was concerned about the accuracy of this kind of diagnosis kit, but this is the wrong because it used the antibody test kit that I mentioned earlier, the second method. So real-time RT-PCR has about 99.999% accuracy. So as you can see here, the Korean government announcement that we cannot, we don't need to concern about the accuracy about this kind of kits. So this is how real-time RT-PCR had been going. First, we use the primarily to replicate the base sequence to proceed with the PCR process. I'm sure you all learned the PCR method in high school, so I'm not going to tell about all of the process of PCR. And if the PCR had been gone, then you will put the probe in a sequence that is uniquely detected by a PCR. And you can measure the signal from probe. If the signal is high enough to say this is positive, then the researcher determine this patient is positive to COVID-19. In fact, in order to utilize primer, you must first determine what genes you're targeting. As I said earlier in the previous presentator's question, the genes position in each country are different for diagnosis are different. As you can see, there are 1, 2, 3, 4, 5, 6, 7. 7 countries have their own gene target. This table, this country is the this table shows the genetic setting of the 7 countries recommended by WHO to be suitable. Right before the class began, I went to the WHO website and found statistics on the phenomenon of COVID-19. So far, about 0.4 million people have been infected, and 80,000 people have died. I hope the earlier diagnosis will help people get treated earlier and be healthy again. Thank you so much.
Okay, thank you very much, Juhan. There may be some other questions we can discuss, but we are running out of time. So let's hear last presenters the presentation first, and then let's do the combined questioning. Okay, Taekjun, are you ready?
Okay. Okay, I will start.
Yeah, go ahead.
My topic is a study on the treatment of COVID-19. And more specifically, I will explain about the structure of coronavirus and the infection route and what can be the target of drugs and the types of vaccines. First, this is the structure of coronavirus. And virus has 4 proteins: spike protein and E protein, M membrane protein, and N protein. Spike protein and E protein, membrane protein together create a enveloped membrane, and the RNA and N protein exist inside of the virus. And the diameter of the virus is averagely 100 nanometers. But the most important part of the virus is spike protein. So I will explain more. Spike protein exist on coronavirus, all of coronavirus, MERS and SARS, and COVID-19. The spike protein composed of 2 parts, S1 domain and S2 domain. First, S1 domain has receptor binding sites, so virus can binding with host cells through S1 domain. And S2 domain facilitate fusion between the viral envelope and host cell membrane. So when S1 domain binding with the host cells receptors, then S2 domain facilitate to fuse between the viral membrane and host cell membrane. Then the infection route will start. There are quite a different between SARS virus and COVID-19 virus. In case of SARS virus, they don't have active sites by furin. And coronavirus 19, novel coronavirus have sites that can be activated by furin. Furin is an enzyme that exists on various of our body cell, such as the liver and the lungs and the small intestines. So it means furin is a key enzyme of our host cell. So many scientists might think that this property of novel coronavirus can lead to they can spread disease faster than SARS virus. Okay, and these could be targets of drugs. Before I will explain this, I will explain about the infection route briefly. The first, as I said earlier, coronavirus is spike protein binding with host cells receptors. That receptors called angiotensin converting enzyme 2. I will call this ACE2. When spike protein binds with this ACE2 enzyme, then the other enzyme involved is called transmembrane protease serine 2. This protease cut off the spike protein. So after that, when spike protein removed, then coronavirus's RNA can penetrate our host cell. So we should understand why these can be drug targets. If we inhibit ACE2, then coronavirus can't bind with our host cell. And when we inhibit transmembrane protease serine 2, either coronavirus's genetic materials cannot enter our host cell. Some scientists use hemostat mesylate. This is an inhibitor of transmembrane protease serine 2. So some scientists research with this material to make treatment. And furin can be other drug targets, as novel coronavirus have an activation site by furin. So I found that these can be the drug targets. But there are no complete treatments yet. And I searched about types of vaccines, what scientists are researching. There are 3 types. First type is whole virus vaccine. This vaccine uses a weakened or dead virus form. This is maybe very familiar with us. And second type is antibody vaccine. It means it uses antibody literally. And there is a new study that using antibody from SARS virus can reduce infection by novel coronavirus. The result is the infection was reduced by 90% by using antibody from SARS virus. And last type is nucleic acid vaccine. This type this vaccine use DNA or RNA, and DNA and RNA, DNA or RNA can inject to our body. Then our body cells make the protein that are encoded in DNA or RNA. So that protein will help the immune system. Thank you for listening.
Okay, a lot of information so you heard. There may be some confusion terms here and there, and there's a lot of explanation may be needed, but let's hear your question first. Any question? Both to Juhan and also Taekjun.
I have a question for last presenter.
Go ahead.
One strategy is, you said, is to block the ACE2 on human body, but isn't there any side effect when you block the ACE2? I mean, there's 2 questions. What is the original function of ACE2 on human body? And second question is, isn't there any side effect?
Actually, the ACE2, I don't know the real function of ACE2, what ACE2 function of our cell, but I found that virus must to use host cell's receptor to enter the host cell and spread their virus. But in case of coronavirus, novel coronavirus and SARS virus use ACE2 for the receptor. So the first question.
Let me intervene a little to save time. Now this the page of the Taekjun's the proposal here. This is a quite different approach of a treatment. Let's say virus is our enemy. Then one thing you can think is directly attack the virus. Vaccine or antibody. Vaccine is the tool to make our own antibody against the virus, or maybe antibody itself to attack the virus. So this approach is directly attacking the virus. That's easy first way we can think out. Now this approach is an indirect way, not directly attack the virus. Instead, we are attacking some of our own machinery, which is working together with the virus. Okay, ACE is one of those protease. It's cleavaging some of those peptides. So the details function is a little too much we discuss here. But any of those machinery, virus utilize for their own benefit. We block that enzyme's function. So that's one possible approach. To do this, we need to understand exactly the virus work. Virus invade our cell, how they proliferate, and how they live from our cells. We need more study of those mechanisms, and we need to check. And if we attack this our own enzyme, whether it may have some side effect or not, that's the next question we should ask. If it's essential the protein for us, of course, there may be some side effect or toxic effect. So that can be next the question we should ask. So for now, let's just think this is another approach we can do, not directly attacking virus. We can use some other approach to modulate or to attack some of our own machinery, our own protein. We can control this. Okay, that much. Let's think for now. Other question?
I have a question for Sir Jung Joo-han's presentation. I'm quite confused with the methods of RT-PCR because the explanation shows that we can be tested as positive if the results show a passage of a threshold. So you have to get certain amounts of those proteins to get to test it to be positive. So does that mean that we have to have enough coronaviruses inside our body to be tested positive? So I thought if we only have one virus, I think we can be tested for positive. But the RT-PCR result mainly assumes that virus have already proliferated. So
okay, before we go into the details, let's figure out. Joo-han assumed that everybody knows about the PCR. Is that true? Everybody knows the PCR? Do you have anybody who does not know what is a PCR? Doyun, what is a PCR?
PCR is replicating of DNA.
Okay, good.
System.
PCR is a polymerase chain reaction. The method is to amplify our DNA exponentially. Okay, what is RT?
Reverse transcripting.
What does it do?
Change RNA to DNA.
Okay, usually in our regular cells, DNA is a template to make RNA, and then RNA is used to make a protein. That's a usual flow. But this virus is, interestingly, the genome is RNA. PCR can be used only for DNA. So this virus should have this conversion backward from RNA to DNA. So RT-PCR means first step is changing RNA into DNA, and then PCR make amplification. So this RT-PCR method is not measuring protein amount. This is measuring genetic information amount. Okay, and this method is very, very sensitive because it is exponentially amplify the signal. In principle, single virus can be detected. If the repeating cycle is high enough, then even a single virus can be detected. So some of those previous other pages, antigen antibody test, this is measuring the protein amount. This method, there is no amplification of the process. If only when the virus has enough numbers of protein there, then we can detect. That's why false positive, false negative is very high. Now this PCR method is amplifying the DNA and detect the DNA amount. That's why this is very, very sensitive. So sometimes maybe 1 copy versus 10 copy, it's difficult to say quantitatively which one has a higher, because after it reaches some threshold, only we can tell yes or no. Still there is some error range, but this method is just tells you that you have a virus or no virus. Okay, too much details, we don't have a time for today. So today we finish here. Then now, as we have seen here, we need a lot of those the basic informations. So that's something that we are going to do from now on. So thank you very much for presenters today. It was very good today. And from next week, we are going back to basic the materials, then more defined study material and also define the questions we are going to handle to build up our basic ability first. So next class will be next Tuesday, 2 p.m. And then there is now the 3 sections of a different the topic, mainly the antibiotic we will start. And then the deadline for quiz is Tuesday, 6 a.m. before on the same day of the class. And there's an assignment, but the assignment today is 1 day later. So you can start early, but you can study together with me on Tuesday, and you can finish up that assignment on the 1 more day, okay? And then, you know, as we said, once if you have a little late work to send me, then what should you do? By the deadline, you send me, you upload that onto the iClass system. But if you feel you are late, then you can do both. To make sure that it reaches to me, then you can. I did not put up any systemic the deadline on the iClass. You can upload anytime, but to make sure I got your material, then you send me to my email together. Okay?
Yes.
So any other question for today? If not, you can accumulate your questions, and then we can discuss other questions next the class. Okay?
Okay.
Okay, guys, good job. Thank you.
Thank
you
you.
very much.
Thank you.